Can you hear me in the back? If I could get a new battery for the microphone, it's going to go out shortly, okay? So I'll need a new battery for the microphone. So we'll get started and then probably have to switch over the battery and the microphone, okay? So a few quick things. As you know, this week we're doing bioinformatics. So I'll run up and grab the battery. So think about the last few slides here where I'm going to try to go over some genetics problems with you. I actually want you to try to work through those. Yeah, the red light. How long does that last? Okay, if the battery dies, I'll do the transfer during lecture. So a few quick things. Since the genetics part in the lab exam, I want to try to do a lot of problems from the lab exam reader. I don't know how many I'll get to tonight. As I told you, I'd like to spend only about 30 minutes or so, maybe even less if I can, on bioinformatics stuff. You can watch the webcast from spring 2010. Don't spend a lot of time on sequencing the new technologies. You will need to know one thing for the lab exam that I'm going to tell you right now. So those of you who are here, you don't need to know the technique, but I'm just going to tell you can take a single DNA molecule, like this, and you can separate it into the two parental strands and add a primer, and you can sequence a single molecule of DNA. So not a population like we talked about before. So you might have noticed when I talked about sequencing, I said it's almost always an identical population of molecules. In this case, you can actually do some techniques where you can sequence a single molecule of DNA. And the way that works is it's not through chain termination. You basically have your DT, DA, DG, and DC with a specific fluorophore. So let's say this one's yellow, red, green, blue. Just made those colors up. If this was an A here, the T would incorporate here from that phosphodiester bond to that primer. The yellow would be attached. You use a reaction to cleave the yellow. You see that flash of yellow light. So you record that as yellow. Then you go through and add the next one. And if it's DG, you take and that forms that phosphodiester bond. It will then take and you do a reaction, cleaves off the green. You see the flash of green. So you can indeed sequence a single molecule of DNA. Okay? You don't need to know the technique, but I am going to ask a question on the lab exam about a single molecule of DNA or several with respect to sequencing. So I just started to go over that. Now, any questions about that? Technique's not so important. Just trying to tell you you can do a single molecule, right? Or two or three. Yes. What's important to know is that if I had a question about sequencing and I say you can sequence an individual molecule, I don't want you to think, oh, there's a million templates in there, and therefore I got to think about a million different templates and how those million are reacting. You can think of, if I said there's 10 molecules that I'm doing single sequencing, then there's 10 molecules. You can think about each of those as being separate reactions and 10 of them, okay? All right? It's okay. Was there a question? Sure, the question is, what do we need to know on the lab exam for this question? My thing is, I want you to realize that if I say I'm doing single sequencing molecules and there are 10 molecules, that means there are 10 molecules that I'm sequencing. So I would have 10 identical molecules that I'm sequencing. And I'm at 2, 4, 7, 8, 9, 10, and you'll my primers. So it's not a million, but it's 10, and I can look at each of these separately. So I can see the results here, I can see the results here, 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 here. It's not like in the past where you had a large population of molecules and 0.3% stopped at the 5th position, and then 0.3 stopped at the 6th position, okay? Does that make sense? All right. So anyway, just trying to clarify that in case you see that in lab exam, don't freak out, oh my God, I've never seen single sequencing, okay? Resources are lots for lab exam 1. I'm going to hold extra office hours. There's reviews online. There's 5 exams in the exam reader. I highly encourage you to do 1 exam for sure by next Monday. Preferably 2. Set aside the time, take it graded. If you get a 85 or 90, you should feel very good about yourself. Because when you take the next one, your scores will keep going higher because you'll see the same thing over and over, just slightly different ways. If you get a 30%, it's not a good sign. Any questions about that? Make sure you do too, because next Monday, I would like you to email me which questions you'd like me to go over from the exam reader. Give me the page number, the question number. I'll take all the emails you give me, and you will design next Monday's lecture. So basically, I will go through, see what you guys want me to cover, try to make it most logical, present those, and then that will go from 5 to 6:30. That will be webcast. And then from 6:45 till 8, I'll stay in this room for additional questions, and that will not be webcast, okay? So I will have equivalent extra office hours next Monday night. Is that okay? So that's the way to try to do more office hours. All right, so you should have seen already the one set of slides in your reader that corresponds to spring 2010, plus my supplement. I'm going to go over. So here's the office hours for this week. Okay, I'll post this. You already got an email. I sent it out through BSpace. Please, when you get announcements from BSpace, try to read them. What you'll see is that I posted this. So we see that there's 1, 2, there's additional office hours here. Oh, I forgot. Brandon's also holding office hours here 12 to 2 on Thursday. So you can add that. I just remember he's doing both Wednesday and Thursday. So I'm holding extra office hours on Friday, a little over 2 hours. On Wednesday, I have 1 plus another 2, so 5 office hours this week. I'll hold the thing next Monday, 5 to 6:30, 6:45 to 8, plus try to hold an extra office hour on Tuesday, okay? So any questions about this? We're trying to help you. Please try to do those exam readers ahead of time so that you have some questions for us, all right? BSpace, you can just go through the various resources that are there in terms of the lab. You've heard the story about central dogma DNA to RNA to protein. So when you replicate DNA, that's replication. PCR is an example of replication, but it's not cellular replication, all right? RNA, to go from DNA to RNA, you have transcription. We've not gotten there yet in the 1A class, but within the DNA, there are things called exons and introns. And so when we take the locus of a gene, so on that DNA, the double strand, there's a stretch, and we call that the gene locus. And we've used this term before. It means the position where the genetic information is. You will read it out as a molecule of RNA, 5' to 3', and there's some processing of it. And in particular, what happens is certain regions are removed from that RNA so that the final RNA product is smaller. So basically, certain parts are stitched back together, and then there's some modification. And that's called RNA processing, okay? And we find that in eukaryotes. So the parts that exit from the nucleus are called exons. The parts that are removed from the RNA and that are not in this final product are called introns. Okay? So the introns are removed. Now, the way the programs display this is they make the exons, the part that exit, they put those as boxes. Okay? So the exons are the ones that are stitched together. All right? So as part of this lab, you're going to look through, check out some disease alleles, information like that, and look for the gene. What chromosome is it on? Which arm of the chromosome? And by that, remember we painted around chromosomes as having this kind of shape in, metaphase like this, where there's that constriction. They talk about a short arm, the P for petite, and the long arm, Q. So when they talk about a chromosome, they may say it's chromosome number 16 in the human, and then they will say it's RMP, and then they'll give it some other information like 18.6. This 18.6 is basically referring to some banding patterns that are seen when this chromosome is divided. And it starts from this position 1, and so 18.6 might be here, and that's where that gene maps to. It maps to chromosome 16, the short arm, band 18.6. Okay? Any questions about that? And I'll show you that information shortly. This illustrates the splicing that I'm talking about. These are the exons. So when we get this first transcript, they sometimes call this a primary RNA transcript. It includes exons and introns. It has a 5' end and a 3' end. And what happens here is this gets modified so that only the exons are in the final mRNA product. This is what will be translated. That's what gets translated by the ribosomes, the structure that makes the protein. And it will have a 5' cap as part of this processing and a 3' poly A tail. And Dr. Patel will have a lot more time to talk about the functions of the cap and the poly A. It helps with stability of the RNA and transport out of the nucleus. The bottom line, this is messenger RNA. It's going to encode for protein. And it needs some way to read this. So what happens is the ribosome will bind. There's a 5' untranslated region. And there's a specific start position within this mRNA that says start translation here. It's an AUG, encodes for methionine. So the first amino acid is methionine, and then it will go on and do this, okay? The 5' UTR stands for untranslated region. The 3' is untranslated region. So there's a start that specifies the start of the protein, and a stop that specifies the stop of the protein. In between, this is read in groups of 3 or things called codons, okay? And there's an entire lecture in 1A about translation, an entire lecture on transcriptional processing. We just don't have the time. This just illustrates the DNA. This shows that there's alternative splicing. In one case, it might be exons 1, 2, 3, and 5. In another case, 1, 2, 4, and 5. So this means that since this is the mRNA, the final protein product may differ between these two transcripts. So they might have slightly different KMs, slightly different affinities, certainly different structure, because the primary sequence is going to be different, but they're going to probably typically do a similar reaction, okay? For example, some genes have maybe 1,000 different variants. That's very unusual. So when we talk about 30,000 genes in the human genome, we mean 30,000 of these, but because of alternative splicing, we may indeed get over 100,000 different protein variants, okay? So it's not great to say, oh, just it's the number of genes that we have to compare. It's really the different number of mRNAs or protein products you can get, okay? Any questions? And then this just illustrates some stuff about how RNA can form secondary structures, and in the 1A class, I'll talk a lot about things to maintain the stability of the RNA. Question? Who has a question? Yes. So the AUG, so if the RNA is like this, 3' end, 5' end, the cap, the poly A, this is the RNA. If this is the translation start site, it would read AUG in the 5' to 3' direction. Okay? So in the DNA, the template for this, the template for transcription would have been 3' to 5', and would have been a C corresponding to there, an A to there, and a T to there. Okay? Is that clear? So oftentimes people say, well, if you know this is the template, the other strand was like this, and if you look at this, this would be an A, a T, and a G. So they'll often say this is the coding strand of DNA because it matches the RNA in the same directionality, except for the replacement of a T with a U. But this is the template. Okay, this is the template. That's what was actually used to make this RNA. But when they talk about DNA strands, they'll say this is the coding strand template because this matches that. Some people say one's a Watson. Others will say the other one's correct for Watson crick. I prefer template. Is it clear that this is the template, that this gave rise to this RNA? And therefore, the other strand in the double-strand DNA would have been this sequence, that directionality, and the coding, because it matches the RNA. Except, of course, for processing, right? All right, various websites we'll be using, a lot of them. The lab write-up is fairly straightforward. It's a matter of just going through, following the steps, inputting the oligopeptide or the DNA sequence that you get from your sequencing reactions, okay? So the way this is going to work for you is if you've started on your pre-lab, you basically give you some information, you work through it, almost identical steps to what you'll do in lab. But for the way you'll introduce this in lab, is we're going to give you a peptide sequence, particularly an oligopeptide, which is a short stretch of amino acids. And this is for human disease. So we'll do most of our blasts, etc., restricted to Homo sapiens, okay? And what we're going to do is give different groups different oligopeptides. So one group may have this one that's 10 amino acids long, and they're going to blast that. And what they're doing is they're comparing that against a database of all these protein sequences, DNA sequences, RNA sequences, etc. And this is on servers, in particular NCBI. And there's a huge database. And when you type in this 10 amino acid sequence, that's your query, because you're trying to look for some information, and then it'll give you back some results. And what we're trying to do is get you to realize that with just a short piece of information, you can go search this database and figure out what protein this was a part of, and what disease is correlated with defects in that protein, if there's mutation there, okay? So that's how you're going to introduce yourself in the lab. This is just sort of running through what your pre-lab worksheet is. We have you start off with this gene here, and then you're going to go through some various web pages and look for, so if you see here, it says this is chromosome here, some information down here, okay? And there's going to be various links on the right-hand side. And this lab is really one of these sort of just go do it, just follow the steps. It's pretty straightforward. The one caveat is NCBI is always updating their website, constantly. So in fact, what I did this morning was I went through their website and went through the new screen captures because it's been changed since I printed the lab manual a month and a half ago. So I have a new file on DSpace and also sent you an email that has the new screen captures. Most of it's not that critical, but sometimes people get confused when the screens don't match anymore. I apologize for that, but they do this all the time. They always update it. In fact, I checked right before lecture, and it's different than what I got this morning. But just slightly, so I didn't redo it, okay? Because they just updated it as they get. In that case, what changed was someone must have put in some new sequence, and as a result, I got a different hit than I did before. So the screen capture won't match yours in that file because now there's another data set in there that wasn't there before. Anyway, when you see this, there's going to be stuff to the right, stuff to the left. There's actually stuff to the right that's really useful. There's various links here, genomic regions that tells you where is it in the genome of the nucleus. Most of these genes are going to be nuclear encoded genes. So when you do this blast in, I don't know if we have any mitochondrial ones, but we could, because certainly we have mitochondrial DNA, and we can have defects in mitochondrial proteins. But I think we've chosen mostly nuclear ones, but I'd have to double check, okay? But I think all the ones we have would be in the nucleus. So they'll give you information about that. Transcripts, that refers to Are there variants when you get that RNA product? Is there alternative splicing? If they list several RNAs, always take the top one. Always take the top one. Whenever we've gone through and made answers, etc., we've always picked the top hit, okay? Of course, that may change between Tuesday and Thursday if we get more data, right? Reference sequences, that just gives you, various things in terms of that. MapViewer helps you see how it's positioned on that chromosome. PubMed, this is a wonderful resource with various literature, articles, full citations, full articles, etc. Links to various things, so that's really useful. The things you can do, it's pretty amazing. And again, this is now just various things. In one case, we asked you to look for homologene, look for homologous genes in other organisms, etc. So that's going to sometimes be hard to find. You sometimes have to scroll all the way down. So if you think, oh, I scrolled to the end, it's actually a lot of pages, okay? And again, it's one of these things you have to do. One of the things you're going to be doing is looking at the RNA, and it gives you a bunch of resources, and at the bottom it gives you this. Now, if you notice this has a T, so is this really RNA sequence? No, if it's got a T, but they can make it into RNA by just switching it, adding a U, right? And what we're going to do is once you have the RNA, you're going to translate it. As I mentioned before, once this RNA is processed and we get the mRNA, it's translated. As we talked about earlier, it's read in groups of 3 or codons, okay? In vivo, in the cell, the ribosome knows, here's the RNA, here's where I should start translation, it uses information, and it starts translation. But when you want to figure out the protein product, and you put it in a computer program, the computer program has no idea if you put in what's equivalent to the coding strand or the template strand, right? Has no way of knowing. So when you translate this, you're going to get 6 different results. So let me just show you why you would get 6 different results. So if we put that sequence in there and translate it, and I'm just going to make something up, ATC, CCG, ACT, and I'm going to say this is 5' to 3', then, actually I need more space, then ACC, GCT, and assume there's a start codon here so that I can have some information. And on down to the 3' end. If I translate this, what would happen is I would say, the computer program would say, I'm going to start this, and there's a codon table, and it will say, that ATC corresponds to this amino acid, and this CCG corresponds to this amino acid, and this one to this. And so it would read them out as amino acid 1, then link to 2, then link to 3. But that's only one possible reading frame for that strand. The second possible reading frame would have been, because the computer doesn't know, would have been TCC. So do you see how that's one reading frame, another reading frame, another. So then I would get a second, so this is reading frame 1. I get a second reading frame, which would have been the TCC. So it would say amino acid 1, amino acid 2, etc. And then, hopefully I have a third color. I would get a third reading frame, which would be the CCC, unlike that. So that gives me 1, 2, 3 possible reading frames for that direction. Does that make sense? Who doesn't see that? There's no fourth one because the fourth one ends up being the same as the first one. Okay? But the computer doesn't know if it was supposed to be that strand, or was it supposed to have been the complement? Was it supposed to have been 5' to A, G, C, and then G, G, T, etc. So the computer will do another set of 3 translations where it will read this way, a second time this way in groups of 3, and a third time this way in groups of 3. So you get 3 reading frames from that one and 3 reading frames from that one because the computer has no idea ahead of time what is the correct reading frame and which strand it should be using. You just type something in, okay? So when you do this, you're going to get results that look like this, okay? Now, they changed the website. So your direction says do not use verbose when you do this translation. They changed it. So this is what it used to look like. And so let's think about this. So here, if you notice, it's 5' to 3', frame 1, frame 2, frame 3, and then it says 3' to 5', because what it did was it said, hey, let's just do, the opposite of that, okay? So let's just run through this. There's 6 reading frames there. Which one do we think is probably correct? Do we get very big protein products here? No. So do you think that's the correct reading frame? Probably not. How about here? Do we get big protein product? We don't have methionine, which is the start. So it boldfaces the methionine to know that's the start. So although it looks like there's a big sequence here, there's no start there. So in fact, does this reading frame give us a big protein product? No. How about this one? Maybe. We got a methionine there, and it goes to this, and then we go there. So maybe that one's fairly long. Let's try this. Methionine, stop, methionine, stop. That one doesn't look very good. We got this, but it's not real good. When we come down here, there's methionine, stop. Probably not that. No methionine. So it's probably this one. Most likely. You can't be 100% certain, because maybe it is a short protein product. But most likely, this is the correct one. So now, you do want to use verbose, because this is what they've changed. If you do verbose, it now highlights for you the longest stretch, the stretches. So it's real easy to see, whoa, that's it right there, because that pink stretch is way bigger than any pink stretch there. Most likely, okay? So they changed that, so verbose makes it much faster. Is that clear? So you want to use verbose there. Again, it will give you 6 reading frames. I only captured 2 of them for the presentation. So make sure you use verbose and not include nucleotide sequence. And it's a change they made. Okay? So anyway, what you can do is once you click on one of these, okay, see how this is highlighted in blue? What you can do is click on one of these methionines, and if I click on this methionine, it will ignore all upstream information. It will pretend as though that was 5' untranslated. So it will say that's the actual translation starts that you wanted. It will say that's my virtual translation because you want me to start there. But then it will print out, you'll see something like this. And then it will give you that information again, and then you can compute various information. Like you can figure out what's the isoelectric point of that protein product, the molecular weight, various things like that. So there are tools you can use after that, okay? Any questions about that? Yes. So isoelectric point means that all these things have specific charges on them because different amino acids will have different charges on that. So if you were to take and put this in an electric field and let it go to its equivalent electric point, it would migrate to a certain spot, okay?